Thank you, Linda. As you can tell, I'm not actually retired. It was just a way to get away from administrative work. I highly recommend it. So I see that we're running a little bit behind this afternoon. As you've been told earlier, the bios of the speakers are in your app. Plus, these 4 speakers have already made presentations at the conference, so you've had an introduction to them. So I'm not going to spend more time telling you about them. But this session, as Linda indicated earlier, is a little different. So what you've been hearing about today are some exciting and potentially promising new methods that people are working with at different stages of development. And so what we're going to do in this session is have 3 of these people talk about the steps in the process of actually doing that development and getting a new product to market, making it commercially available so it can be put into application. But in keeping with, or keeping in mind throughout, the regulatory guidelines and sometimes hurdles and challenges that we have to face in doing that. So Linda Rhodes, of course, she's our go-to person for that. She'll be stepping in the sequence and telling us, warning us about some of those. So I think if I do this, yes, then we... I have to put my glasses on. Yes, so this is the order that we'll go in, and supposedly these talks are linked together, so they will flow seamlessly from one to the other, but I will still stand up each time and prompt the next speaker to come up. So Lindsay Van Zandt will be our first, addressing doing the first stage of research in the lab.
Here we go. All right, thank you, Sherry. As she mentioned, I'm going to focus on actually doing research, and of course, I think you guys have figured out by now, I'm kind of a cat person, so this will be how we do research in cats. I just want to mention really briefly, too, so this is home for us at the Cincinnati Zoo. We have our research facility right on zoo grounds. I just want to briefly mention kind of our overview research, kind of what we focus on and how we fit in here is, first and foremost, we do use the domestic cats as a model for the other members of the Felidae family. All cats evolved from a similar size ancestor, a medium-sized ancestor, and so their physiology, especially reproductive physiology, is well conserved. But in addition to that, I have a strong research interest in shelter medicine. We know stress is bad. We know there's lots of aspects of shelters that are stressful to cats, and so we spend a lot of time looking at how do we reduce stress in shelters, and similarly, how do we, with that, reduce disease transmission? We do a lot of work looking at operant conditioning and how that can promote a better bond and help. And then, of course, we also are interested in how to humanely reduce free-roaming cat populations. So this is how we house our cats. Right now, I think we have 85 cats in our research colony. By and large, most of them are group housed. We do this for a few reasons. Is one, it just gives the cats more space than having to individually cage them. They have more space to move around and express their natural behaviors. And also, our cats in general are quite social. You know, we sort of think of cats as being very solitary and aloof, but I think it's very enriching for them to get to have these relationships with each other. There are a few exceptions. For example, of course, after surgery or something like that, we would have them caged for a few days. And our males, by and large, are individually housed. Some of our males do get along and do get to be together. Sometimes they decide they no longer want to be together, and we have to make adjustments from there. You'll notice in the room, too, this is a typical room, so lots of shelves. Our cats, of course, love height. Little hidey holes to hide in, different toys for enrichment, and, of course, lots of litter, lots and lots and lots of litter for these guys. We do have one room that doesn't have any shelves, so whenever we do take in new cats, we do individually house them for that period or group house them in smaller units. And so we have to come up with more creative ways on how to give them height. Our colony manager is getting really cute with these little houses she's buying. We just got a pirate ship, you can see. So the next step, of course, is to get a cat pirate costume, I think. So hopefully by the next one of these talks, we'll have a cat dressed up for you guys. And And so so I'm a veterinarian, Bill's a veterinarian, Bill's postdoc is also a veterinarian. So between the three of us, we share the coverage for the cat colony. There are rare times, which is going to be later this week when Bill, myself, and Raquel are at a meeting, that none of the veterinarians are there, but that's one of the advantages to being at a zoo. They have 3 full-time zoo veterinarians that are perfectly capable of handling any issues. So it's nice to have that ability. So we have a small treatment room back in the cat colony. So for very simple things, ultrasound, blood draw, simple wound repair, those sorts of things we can do back there. But of course, for any sort of surgery, we do have a full surgery suite, which our nice little tiger patient is demoing for you guys. So let's see, we don't want to go. So we also spend a lot of time doing training and enrichment and socialization. So our colony manager is fantastic. She scours the internet looking for new toys. And here you can see one of our orange boys really enjoying Cat It's, the line of these toys with different textures. But we do spend a lot of time getting our cats socialized. We want them to be happy around people. We want them to enjoy people because ultimately our goal with our cats is for adoption. And so it depends on the type of study we're doing. We always have the ultimate goal of adopting out our cats. It just kind of depends on whether they have a really easy study where all we're doing is feeding them dye and collecting their poop, or if they're involved in a study that needs multiple blood draws, or we have to do surgery. Those are kind of the decisions we make where we say, okay, you've done your due diligence. Now we're going to find you a home. So actually the 2 cats here on the left are actually retirees from the Gonacon study, in their happy home. And actually the cat in the upper right is my cat I adopted from Crew. So I always joke, it's like that commercial, I'm not just the hair club president, I'm also a client. They are wonderful cats. I don't think anybody that works at Crew, very few people that work at Crew don't have a Crew cat or 2 or 3. So yes. And so yeah, there are many people here. Linda has one of our cats, Amy's sister. So yeah, Camille is very happy. Ask Camille about her cat. Everyone's very happy. Anyway, so we love our cats. We love getting them into homes. Just to tell you guys a little bit, hopefully I don't think I'm getting into Linda's regulation part, just the regulation of what we have, for those of you who maybe aren't as familiar. Under the purview of the Animal Welfare Act, the USDA actually comes and inspects and regulates our facility. And so basically their goal is to, again, enforce that Animal Welfare Act. So they come to make sure everything is kosher and we're treating the animals by appropriate standards. That's anyone that falls under this category of using animals for research, teaching, testing, or experimentation. And so annually, they do show up, and they can actually show up more often than that if they so desire. And it's a surprise, so that's always a fun day. It always seems like it's when we're in the middle of doing a bunch of procedures is when USDA shows up. But they come and they look at our records, our practices, they come on premises, and all that good stuff. And you can actually look all of these up. So here's a nice report card from Crew. We tend to do very well. So you can actually look this up online and see how any USDA-registered research facility did. Additionally, we have what's called an IACUC, or Institutional Animal Care and Use Committee. And so their job is to actually review the research protocol. So before we can do anything, this has to go to this committee, and they actually have to make sure, again, that we talk about the 3 R's. Like, do we have to use animals? Is there any way we don't need animals for this study? Or with reduce, maybe we don't need to do this in 300 animals. What's the minimum number you need? And refining is looking at the ways to make whatever you're going to do as most animal forward and least stressful as possible. And these folks actually also come and they do inspections twice a year and come inspect the facilities with that same idea in mind. Now, additionally, if we are using a study that has some sort of recombinant or synthetic nucleic acid, so when we talked about a lot of gene therapy today, any of these gene delivery type studies, we have an additional layer with an institutional biosafety committee or an IBC. And their role is not so much the welfare of the animal as just the safety of the study itself, considering risks to the environment, considering public health risks. And so their role is more, again, to sort of just provide technical advice on these safety procedures. They also periodically come and inspect our labs. So we have a lot of people that come through our building every year. And helping us develop emergency plans should there be some sort of contamination, some sort of spill. That way we're proactively thinking of this from the beginning. And I'm just going to end by a few of the challenges we have. One is that cats are not big mice. So oftentimes when we're thinking about these agents, the numbers that people who primarily work on mice, they're afforded large numbers in their study groups, and and so what's biologically relevant or statistically needed is sometimes very different than what's logistically possible. And again, for us, we're adopting all of our cats out at the end, and the idea of just how we're going to house these cats. And they are quite sentient beings. They're quite clever. We ask a lot of our keepers to maintain these cats in a happy environment. So for us, we're not at our maximum carrying capacity. Technically, via USDA rules, we could have a lot more cats in our facility than we do, but we're happy with the number we're at for how good of care we can take right now. So it's always a challenge when people want to do these larger studies of how many we can do. Similarly, cats are not little dogs. So there are endpoints that researchers often want that don't sound hard. Things like rectal temperatures. It's possible in a cat, but that's going to take some training. Things like, by rectal temperature, I mean like having to do that like every day or every week or something like that. Similarly, even things like palpating where there was an injection site. Obviously, that's really important. When we think about some of these agents, we need to know if it's there or not. But that again, cats are very different from dogs, and the mere idea of you trying to do a simple restraint or trying to just palpate can be quite challenging and takes training on the front end. They do have unique reproductive physiology. One of the biggest is this whole spontaneous versus induced ovulation. When we're monitoring animals hormonally, it's really quite nice to have estrogen followed by progesterone because it's a nice definitive, they're cycling, they're ovulating, or they're not. With cats, they might spontaneously ovulate, they might not, and so it gets a lot harder when we're looking at treatments to say, okay, she's not ovulating anymore, but again, that's a normal cat, maybe. Graphic picture coming up, so if you don't want to see it, close your eyes. We get a lot of pyometras and you don't see this in private practice with cats because most people that have cats are going to spay them because an intact female in heat is not fun. And most private folks who have these intact cats tend to be breeders. And pregnancy is quite curative for a reproductive tract. So we're not, of course, always breeding all of our cats. And so, especially the ones that spontaneously ovulate, we do see this come about. The good news is I think we're really good at these. Our cats do really well. We have a nice treatment plan in place and they do well. But again, it's just another struggle that we face with having these cats intact and not breeding them regularly. If you close your eyes, you can open them. The picture's gone. And then I'm just going to end really quickly about thinking about adoption, particularly with these gene therapy cats. The FDA does actually consider them genetically engineered. Whether or not you're integrating anything into the genome, they're still falling under that purview. What's actually really great news and surprisingly rational decision is that this is our sort of benchmark is glowfish. You guys familiar with glowfish? You could buy them at PetSmart. They've been genetically engineered to glow, to make these really bright colors. And the FDA is completely fine with them, which is shocking and fantastic. And so in theory, our cats would be very similar. They are not part of the food chain. They really shouldn't be posing any threats. And actually, our collaborator, if you were here earlier, for the research I discussed today, Bruce Hay, actually has an email from somebody from FDA saying that, in essence, these cats should be able to be adopted out. So that's good, but we still haven't adopted any of them out yet, and so it's still kind of a concern that we think about. And that's it for me, and we'll just pop along.
Thank you, Lindsay. So that was our lab research perspective, and next will be Amy Fisher, who will take us into like the clinical.
So, hi everybody. I'm going to share with you how we created a hybrid lab field trial environment, and many of you saw this yesterday. So I'm going to speed through the slides you've already seen and then share a little bit more because we have a little more time to get into it. But what inspired this work, ACC&D always was really intrigued at the success that Julie Levy found in the lab. the potential success for Gonacon as a free roaming cat contraceptive. And so we really wanted to see if we could replicate those results in the target population of free roaming cats. We couldn't go quite to the field trial stage after the laboratory, so we set up this hybrid environment somewhere in between, using cats that were more representative of real cats. So we had a genetically diverse outbred cats from random sourcing, and we created an environment that was more loosely controlled than the laboratory. So briefly, we put together a population using random sourced cats from a variety of animal control agencies and a few other sources throughout Illinois. And we brought together a study population of 30 females, 20 of whom were injected with Gonacon, 10 with saline, and then we rotated through 5 breeding males. And I don't have to get into a lot of this because I know Joanne expertly shared the study this morning. But just to tell you a little bit more about how we created the environment, we put together a pole barn and expertly created the a very unique environment with lots of places for cats to be cats and do cat things inside. And we had this space within the climate-controlled pole barn, but we also had a space outside. We enclosed about 1/3 of an acre so that we could provide the enrichment and the exposure to the natural photoperiod and that sort of thing. Again, trying to create a humane environment that was a step more toward a free-roaming cat population instead of a tightly controlled laboratory setting. Our situation was such that we were a USDA licensed research facility, so we did set up an IACUC just like Lindsay described. We also were advised by our State Department of Agriculture to license as an animal shelter. In the state of Illinois, that was a structure that worked for us to be able to transfer cats from animal control agencies and then adopt them out when the study was over. This is not a situation that will be relevant in every state because each state has different legislation pertaining to animals, but this is what worked in Illinois. A few lessons that I shared yesterday, the first 2 being things we would really do better next time. I think when we started this study, we had this romantic notion of this glorified barn cat population who are just going to be happy and have this breeding trial. And we didn't really take seriously enough right away how stringent we had to be about shelter medicine principles, about training, about having a proper quarantine and isolation area in the pole barn. We didn't have that. And we also had substrates that couldn't be sanitized, and so that was something that when we do this again, and I hope we do, we'll certainly fix those things. We also severely underestimated the amount of expertise we'd need from our veterinary friends. We planned and budgeted for our experts, our specialists, to come in, like Lindsay, who came and measured testicles and did our fertility tests on our males, and our radiologist who did our ultrasounds, but just in terms of the day-to-day stuff that happens when you bring 35 cats together, we just didn't have enough consistent veterinary support for that, and so that's one thing we'll do better next time. 2 things that we did really well. We had an extremely detailed protocol, and again, I thank Linda for this. We really sat down and hammered out a very long protocol up front. We had forms for every event, for every cat. Each cat had a thick binder. We were so organized, it was very impressive. And we also had, just a chain of custody form, again, documentation for every single step of the process, which before we started, those of us who'd not done this sort of thing before really didn't appreciate how necessary this would be and how grateful we'd be for it when things were quick moving on the ground and we just needed to grab the right form and go with it. Finally, the one lesson I'll share that, again, I think we did really well, and along with what Lindsay described, we really want to have optimized enrichment for animals in our care. And so we, again, provided nothing expensive or crazy, just a lot of fun stuff for cats to be cats. We had olfactory enrichment. We had a lot of student interaction. But along with the interaction, we also had a strict training protocol where we had our students come out and get our cats accustomed to target training so they could easily be led throughout the facility. They got comfortable being laid back in the tray for the ultrasound exams. They got somewhat more comfortable with blood draws. And so we just had a more relaxed cat going through these procedures, which was really humane, or really important for us to know that this was a humane study because these cats were not stressed. This also really paid off when it came time for adoption because these cats had been introduced to so many novel stimuli throughout the study that they really adapted well to their new homes. And the training also helped us to get them used to going in carriers, going in cars, wearing their little harnesses because we wanted them to have that outdoor enrichment even if they were going to an apartment building. So that's quickly what I shared yesterday, review for many of you, hopefully. A few other things that we can get into today. The behavior observations were just fascinating. Certainly, as part of our study, we recorded all of the estrus behavior that we observed, as well as any male interest in a female, as well as mounting and mating. Everything that we observed got recorded. And as I mentioned, Lindsay came over and did fertility testing on our males, so we knew that every male in our study was a fertile male. That said, we were very surprised when we started the breeding trial portion. There were variations in male interest and performance that many of us just didn't expect. Some males had no interest whatsoever in mating with the females. Some had tremendous interest and were very good at it. And little Wolfie on right here, he had tremendous interest, but he had no idea what he was doing. And it was actually kind of frustrating after a while. So this was just surprising for someone who's, my whole life, I've really just been fascinated at this species and its incredible reproductive capacity. I assumed every cat would be really good at it, but that was not the case. So that's one observation we made. We also saw some strong mate preferences, which I don't know if there's anything scientific to this, but we definitely had males seek out and almost stalk certain females. And fascinating, I think this is just coincidence, there was a lot of phenotypic similarity. So Captain Obvious, the gray and white male, really sought out Hermione, the gray and white female. And likewise, Conway Kitty and Bon Bon also had a similar appearance. So these were our two, I know it's crazy, it's not scientific at all, but we had these strong mate preferences that they showed. We also observed post-spay aggression problems. And so we, as I mentioned, we brought these cats in from animal control facilities. We had a very harmonious population of cats. We introduced males 4 months after we assembled the females. We still had no problems. It wasn't until after the spays that we started to see aggression among our population. And the aggression was initiated by the spayed females. So there's not a lot in the literature to help us understand that. We don't know if it's because of pain or discomfort associated with the spay or because of the abrupt hormonal change by spaying a pregnant female. But there's certainly something to look into there in the future. We also adopted our cats out. Like Lindsay said, I'll go quickly through this just because it was kind of a challenge for us. We had no experience in adoptions. We did find homes for our cats in 6 different states, however, and I'll just briefly go through some fun pictures that our adopters sent us. But that was, you got your costume, Lindsay. But that was a part of our study as well. So just to close with some other considerations, the sourcing, again, how we did it in Illinois won't be how everyone can do it. It's going to depend on your state legislation. Working with the many different animal control agencies, you do have a variation of risks in terms of disease transmission and the backgrounds of the cats coming in and the expertise at the shelters. So that's something to keep in mind moving forward. We did have quite an ethical, we did have quite an ethical discussion about doing pregnant spays. Some on our team wanted to let the animals go to term, and certainly there are points to be made with that. But at the end, we would have had 111 kittens on the ground had we let that happen. So in central Illinois, that's just not a population addition we needed. The adoptions, as I mentioned, they were a struggle for us just because that was an area of expertise that I don't feel we really had. So I can talk to anyone offline about that. And just achieving that hybrid model balance was interesting because you have this tension, you want to create a humane environment, but you have certain parameters of your study you need to meet, and you also want to, be able to collect the data. So there was just this constant tension between the different stakeholders, but I think we pulled it off in a good fashion. And we just published the behavior and other ethical pieces related to this study in Journal of Feline Medicine and Surgery last week. So thank you.
I'm just hoping when we get to the question and answer session that Amy has time to tell us something about how they named those cats. So both of our presenters have alluded to regulatory issues that they had to face, and so we're going to tackle that head on right now, and Linda's going to give us an overview introduction.
So the regulatory piece that I'm going to talk about is very different from what Lindsay and Amy had to deal with, where the cats are really considered kind of a laboratory in a laboratory. And when you need to when you feel confident that you have a product that has a real potential to be commercialized, you have to do what's called a clinical field trial, and that's what I'm going to talk about. So, what is clinical research? In human medicine, it's very straightforward. All the work you do in dogs and primates and rats and mice is called preclinical. And then as soon as you move into people, it's called clinical research. But in animal health, it's hard to make that distinction because we're doing our clinical research in the laboratory, in the hybrid model. These are all done in what we like to call the target animal. So there's not that real demarcation of clinical. So in animal research, in animal drug development, what we call clinical research is when you move into client-owned animals. So they're actually you're going to be testing your product on somebody's pet. And you don't do that until you have pretty extensive safety studies, until you've really defined what your product is. And this is kind of the last stage of what you need to do to prove safety and efficacy. So you're not going to put anything in my cat that hasn't been tested extensively in laboratory cats, because I'm not going to feel comfortable with that. So you have to have some real background on safety. And why do we do clinical research? Well, a lot can be learned from looking at pets in their own home environment. You have to really study an outbred animal of a variety of breeds in a real world situation. And there's just two basic things we learn from clinical research. First, is the drug effective in the population it is intended to be used in? And second, what's the safety profile in that population? And regulatory agencies worldwide require these types of clinical studies to be submitted in support of regulatory review. So just a couple of things to point out as differences between academic type research and what we call kind of regulatory clinical research. So many clinical research studies are done in an academic setting, and they're not as closely regulated as studies required for regulatory submissions. And this listing gives you some idea of the differences. So regulatory studies have to generally be bigger. You have to do them at multiple clinical sites. So for example, you go out and train veterinarians. So I might have a big veterinary clinic in Los Angeles and another one in Chicago and one in New York and one in New Orleans and one in Kansas City. And they're spread out around the country, and each of those veterinarians that run those studies have to be trained in the same protocol. So you have to have a variety of clinical sites because the regulatory agencies want to see that there's not something special about that clinic in New York that did the study in a particular way that made your drug look good, but if you did it in 6 different sites, it wouldn't look as good. So you have to spread out. These all have to be prospective studies, so you can't look back at data. You have to create data going forward. And then I'll talk a little bit more about what GCP means, but it stands for Good Clinical Practice. And there's significant oversight. And then, of course, these regulatory studies are submitted to the government for review. One of the things that they're worried about is fraud. So you can imagine that if I'm a big drug company and I'm paying you as the veterinarian to do this study, you might want the study to come out positive because then I'll give you another study, the next time I do a study, and you'll get paid for that. And there have been instances of fraud in these clinical sites. So one of the reasons that all of these regulations are put in place is to assure the integrity of the data, so that there's no possibility for those data to be fraudulent. So who is who in a study? So many clinical research studies have several different kind of stakeholders. And the first is the sponsor, and that's the sponsor is the company that's developing the product. And the sponsor is responsible for writing the protocol, finding the clinical sites, training all the personnel, providing the drug, doing some oversight of the study, and doing all the statistics. The veterinarian is the person who is recruited by the sponsor, and the veterinarian has to follow that protocol. So in other words, if I get the study at my clinic and I say, well, this is really interesting, we're taking blood at week 2 and week 8. I want to take blood at week 6 also. No, 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 no. You don't do that. You follow the protocol, and you follow it absolutely to the letter, because the regulatory agencies want to know that the study is being done exactly the same at different sites. And if you generate additional or supplementary data, it's going to be very hard to interpret. It could raise all kinds of questions about something you found in that extra data point that might not be consistent, and it's going to really spoil the study. So you follow the protocol. It's your job as the veterinarian to recruit the patients. So when a person comes in with a dog that has the condition that you want to treat, you explain the study to that owner. The owner has to consent, obviously, and this is the kind of the IACUC function, where we have to get a owner consent that's approved by the FDA. So the owner consent has to explain all the risks, all the problems, all the potential downsides, and get a signature from the owner that they understand that and they're willing to enroll their pet in the study. So that's a really important part of the study, is the veterinarian plays the role of recruiting patients for the study. Study design is pretty consistent across these clinical studies that are done for regulatory reasons. They're randomized, placebo-controlled, masked, or other people call it blinded. You have to have this owner consent that I spoke about. Most of them are fairly long-term, because you want to study the effects of your drug over a period of time so you can really assure that you understand the safety profile. And as people have spoken about this previously, that you have to use the final commercial formulation that you're going to actually plan on marketing. So it's a little bit tricky to do this for a contraception, because if you think about it, if I have a product that I want to prove that it's effective for 5 years, I'm going to have to do a 5-year study. And I might even have to do a 6-year study, because the FDA might want to know, well, what happens at the end of the treatment, and do the animals come back into fertility, and are there any safety issues? So defining the patient population that you use is tricky. So I've just kind of, let's use a study for a new long-term contraception as an example. So the study population would likely be pets. Dogs and cats would need separate studies that owners might present for surgical sterilization. And the protocol would say the required age, and that would depend on the approach. The study might specify, for example, that we need male dogs over the age of 3 months. I'm kind of making up a study here. And you would want to specify the fertility status of this animal. You would want to specify their health. You would want to specify what other medications they were on. You might not want to enroll a dog that was on thyroid medication or some other hormonal medication that might interfere with your treatment. So you define all that kind of stuff up front. And then you have to do your long-term study. And I've been thinking about this for a while, because we are really hoping to have long-term approaches. And I think that it's very hard to achieve a long-term clinical trial, because say you need to prove this in 150 animals. If you want to have 150 animals at the end of 5 years, you have to enroll 400 animals at the beginning, because owners are going to move, they're going to get tired of the study, and they don't want to bring their dog back. The dog's going to get hit by a car. The dog's going to get really sick with something else and have to be taken off the study. A 5-year study is a tremendously difficult undertaking. So I think what will happen is that the companies will say, let's have a 1-year label. So let's have our claim be, this suppresses fertility for 1 year, but we'll start with 400 dogs, and we will try to follow them for as long as we possibly can. And for the gene delivery approaches, which might be a lifetime, we will say we're gonna have a 1-year claim. So we will say this will suppress fertility in treated cats or dogs for 1 year. And then at the end of 1 year, we will continue to study those animals. And at the end of 2 years, we will submit that additional data, and we will say, now we're extending our claim to 2 years, so that we can start marketing the product with a 1-year claim. And the label would probably have to say something like, return to fertility may not be established. So that the owner knows this might be permanent, we just don't know, but at least it will get you 1 year. And then the owner will come back to the vet at the end of that 1 year and say, oh, she's still contracepted. Okay, well, that's nice. Come back in 6 months, we'll have another data point. At the end of 2 years, you gather up all those data, you submit that to the regulators, and now you have a 2-year claim. And you do that every year. And I think that's how these drugs are going to have to be approved over time. It will be a very labor intensive. It's a long-term commitment for the pet owner. It's a long-term commitment for the veterinarian. But I think that's going to be the major challenge in getting a long-term contraceptive approved. So once you gather all those data, you write a final study report, you're a statistician, reviews that final study report, and then the FDA will come and inspect your clinical sites. And your clinical sites have to maintain their raw data, the original place they wrote down the information. And the FDA will come and interview the veterinarian and make sure that veterinarian actually did the study, that they have case records for every animal, that they have a name and address of the owner. And so they're looking for fraud. They want to make sure that things are not fraudulent. And once they do their inspection of the clinical site and they review all the data, then they can tell you, yes, okay, we accept this study as a good regulatory study. So it's a very rigorous process. And as the person who taught me how to do GCP said, there are several layers to ensure the integrity of the data, but also to ensure that if 15 years from now, if everybody involved in this study goes in a rocket ship to Mars, anyone can look at the raw data and the final study report and understand exactly everything that happened in that study. When the drug was shipped, under what condition was it shipped, who signed for it, how many doses were shipped, how many dogs were treated, what were their breeds, what were their ages. So if something comes up 10 years from now, 15 years, suddenly there's a big safety problem with these animals. A whole bunch of animals start getting kidney failure. They can go back to the original raw data and they can figure out what happened. Very, very important because, people move jobs and labs change, and everybody who knows about keeping laboratory records knows that things kind of get lost. And this is a way to ensure integrity of the data of these very, very expensive studies. So a study like this that would be a 5-year study would probably be about a $5 million investment. Generally, clinical studies for things like pain drugs or antibiotics cost between $1 million and $1.5 million to run. So these are very, very expensive and rigorous studies. But once you do one, you get a definitive answer. And I think that's what's so powerful about this kind of clinical research. So thank you.
Thank you, Linda. And last but not least, of course, Dr. Rowan is going to talk with us about the next stage, which would be moving into the field, but that then carries its own regulatory hurdles, which he'll address as well.
Thank you very much, Sherry. Thank you. Wonderful set of presentations, and I'm afraid that I'm going to be certainly least at the end of this thing. Joyce had to rap me on the knuckles and get me back on program because I wasn't quite focused when I should have been. And so I've been thinking about this whole process as we've been going through the last 2 days. And I do want to give just a little bit of background of my situation. Yes, sorry. I've been working in the animal protection space since 1976. 42 and a half years now and counting. And most of that time, one of my duties has been to promote the idea of alternatives to animals in biomedical research and testing. And so part of what I've been thinking about in this thing is how do we get these products on the market in ways that don't involve a lot of animal distress, a lot of animal euthanasia, a lot of animal cost, shall we say. And what we heard from Lindsay and Amy, I think, is one example of what we can be doing in terms of avoiding animal costs. So the Humane Society at the moment is arguing that we should be doing research that doesn't cause harm to animals. So we're a little bit wishy-washy on the notion of euthanasia, But one of the main sort of issue is animal suffering and animal distress. And I think most people would agree that the cats that live at Cincinnati Zoo or that lived at Amy's facility weren't really suffering. And this is the type of thing that I would argue is the type of research that we would be able to support. And the Humane Society has challenges in this regard. We've been supporting research on PZP for 15, 20 years now, and or studies or management field trials, if you will, on PZP, and that's porcine zona pellucida vaccine. And we've been doing this in horses and in deer and in elephants. And so basically, and also they've been used, as Sherry can tell you, in zoo animals. The PZP's been used there as well. And basically, it's all field trials. None of it has been basic research, as it were. It's been, let's go out and use this. So Jay basically decided that he was going to apply PZP from the beginning. So he went out and started applying it, showed that it worked in horses. By the way, if one is worried about the welfare of horses that were used in the Assateague Island ponies that were part of the first field trial, the average age of death of those ponies, female ponies, was 6.8 years if they weren't contracepted. If they'd been contracepted for 2 or more years, their average age of death was 21 years. And when they started the trial, not a pony on the island was over 15. There are photographs now of Taffy and a couple of others that are 35 years old. So they're going to die when their teeth fall out. So I mean, there's this huge welfare component behind these particular field trials. The elephant stuff is also all field trial, based upon field trials with elephants. Interestingly enough, it's an immunocontraceptive. And in elephants, I think there's been one failure in multiple thousands of doses. So it's been tremendously successful for an immunocontraceptive in elephants. And so we've been doing all of this stuff in field trials and not going through the type of rigorous controls that Linda described, which is probably going to be required by regulatory agencies for the dog and cat contraceptives or sterilants. So but even there, the situation is funny, because when T61 was brought to the market by hosts, this was the euthanasia agent, the only clinical or preclinical trial that was required was a study done at Fort Collins at Colorado State that involved 24 beagles that were instrumented, and the T61 was the experimental drug, and 1/3 euthanasia dose sodium pentobarbital was the control. Not sure why they used 1/3 the dose. And that was acceptable at that stage for the licensing of a euthanasia drug. So there have been huge changes that have been occurring in this field. And so one of the things that I'm going to be arguing is, what can we do so that we can bring products to market that are going to be safe and effective and not involve a lot of deliberate animal experimentation and the killing of animals. So we've been doing these clinical trials, as it were. Basically, we've been providing management options. So most of what we did on Assateague was a clinical trial, but with an aim towards management. And now the Park Service is using it to manage the population of ponies on the island, come down from over 200 to 85, which is below their management level. It took 25 years for that to happen, because the ponies lived, they stopped dying at 15 and they lived to 35. So what you find happening here, of course, it depends heavily on the agency that is overseeing the product. So the companion animal arena is FDA, dog and cat products. USDA are food animal products, and the EPA is regulating pesticides. And we as the Humane Society had to sort of hold our nose and say, okay, PZP is a pesticide because wild horses are quote-unquote pests, and so we'll register with the EPA. Whether that would be okay for feral cats, I think there are communities that would be very unwilling to hold their nose and call feral cats pests. But you've got Contraceptive, which is this rodent fertility control agent, over-control in birds, and the feral cat issue is something that one has to sort of worry about. So the Phagostone, the USDA lab up in Fort Collins, which developed Gonacon, they published sort of the oversight of what would happen. And interestingly enough, they did very little in the way of field trials. They did a couple of field trials, but mostly what they did was lab work in Fort Collins. Whereas we, looking at the horse product, the PZP, did mostly just field trials and very little lab work. So, and we both got our agents registered at the EPA. So the EPA did not require to provide formal GLP efficacy studies. We were able to use the results from all of our efficacy research, ACC&D, NIST, etc., that was conducted to address various unknowns related to field application. And they produced this positive outcome and that huge increase in longevity in the ponies. That data was acceptable to the EPA. They wanted some safety data, some environmental impact data, but by and large, they weren't terribly engaged with the efficacy trials. If you have to register with the FDA, I'm pretty sure that we're going to need much more extensive GLP efficacy trials. One of the ideas here is that perhaps we could look at what the pet food companies are doing for pet food. Procter & Gamble bought IAMS. Shortly after they bought IAMS, IAMS was infiltrated, or one of the sites that it was using was infiltrated by an animal group who embarrassed the company. And so they set up an international advisory group, and I was one of them, to advise them on how they could move forward. And basically what they ended up doing was they ended up doing most of their research in pet owners' homes. So they recruited volunteers, mostly Procter & Gamble and IAMS staff, to start doing studies, palatability trials for the most part, but also more sophisticated, more difficult trials. And because they were doing it in people's homes, it was also more expensive, because it just took a lot more time and a lot more effort. So there was a cost involved in switching from the standard laboratory trials that they were doing in their facility in Dayton, Ohio, compared to going out into people's homes and collecting the data that way. So these are the sorts of options that we might be able to think about. So non-animal studies. One of the things that I'm surprised by, but gratified by, is that I never thought that in my career I'd be able to say that animal testing could be eliminated. But in 2016, Francis Collins, the NIH director, told the Senate Appropriations Committee that he predicted within 10 years we would be doing most drug studies and drug safety evaluations in non-animal systems. So it's not just, I'm obviously gratified by what he said, but I'm really very excited by the technologies that are coming on. Now that's different from what we're talking about here in terms of sterilants and contraceptives. We're going to have to do the animal studies at some level or another. And the question is, how can we do that? But the animal testing is going to be changing. And it's going to be changing because it's economically expensive, because its predictivity is not terribly good for humans, and because the new technologies are so much faster and quicker and cheaper than the animal stuff. So there's going to be some sort of change happening in the regulatory arena. Not sure how quickly it's going to happen. Collins predicted 10 years. I suspect I've been predicting 10 years, but I suspect both of us are over optimistic. Because regulators are not given many benefits and credits for being innovative. So I think it's going to take a while, but the sheer economic advantages of the non-animal systems is going to drive this forward. So real potential, I think, to use clinical trials, animal trials and studies in which the animals are not harmed, and adopted out at the end of the study in place of standard GLP tox and efficacy trials. As I say, similar approaches are being used in wildlife, in pet food studies and in wildlife fertility control, and hopefully we can have the ability to do more and more of these studies in ways that will address the safety concerns and the efficacy concerns, but will not end up producing harm to the animals. So that's my sort of 5 cents worth here. Thank you very much for your time.
Thank you, Andrew, and I'm sure you agree with me that that was worth a lot more than 5 cents. In fact, I had thought of asking if Andrew would provide at the end of his presentation, he did it at the beginning, his perspective on humane aspects of this kind of research, because he's in a unique position, I think, of all of us. He's been thinking about this and working with it for a long time, so I'm sure that people other than me appreciate hearing that. So we're going to open for questions now. And we've got till 3:30, so a little more than, 15 minutes or so, 15 or 20 minutes. And this also includes the panel members can make comments, ask questions of each other, because this is supposed to be a discussion, not just questions from the audience. So anyone who has questions, if you want to, as we've been doing, come up to the microphones. Starting with Michelle.
This question is for Lindsay. Did you consider using an oral thermometer instead of not oral thermometer instead of a rectal thermometer? We use those most commonly in practice to take temperatures from cats, and they are considerably less objectionable, object to that.
That's okay. I'll just come
Yeah.
I can move too. Yeah, I guess historically I've had those and I don't find they're very reliable. Maybe we just need to try another one. But sometimes I find rectal temperatures aren't terribly reliable either. Just kind of depends on the rectal tone of the cat. So I find sometimes we'll take a temperature and the cat will be like 97 degrees or 98 degrees is what we get. And I know the cat is not 98 degrees. So yeah, my own cats, I have a oral thermometer at home that they tolerate. But I mean, we could for sure. I think some cats still object to ears as much as other cats might object to their rectum. So they just object no matter what we do.
Yeah, no, I think, I mean, obviously there's going to be some cats that object to no matter what you do to them. But as far as conditioning, it's a lot easier to kind of scratch this ear and take a temperature from this ear. And I agree that I found that the oral temperature tends to be lower than perhaps the interrectal temperature is. But if you were measuring, if that was what you were using for baseline,
right,
and you were using that all the time, then you'd be comparing, oral to oral and not oral to rectal.
Yeah, no, that's a good point. 100 points. Okay, am I okay to sit, or is another one for me?
I'll give a couple more for you.
I'll just stay.
So you made a comment about a couple comments. One about cat breeders that it's easy because they're just breeding the cats, so they don't have to worry about pyometra. But actually, cat breeders, and I work with a lot of cat breeders, and I actually work with more cat breeders than I work with humane societies, and they are extremely interested in a reversible contraceptive, so, I mean, your cat breeder market is not going to be your largest market in the country, but we've talked about lots of different contraceptives that are available for dogs. In fact, we can still use mebolorone or even megestrol acetate in bitches used for breeding for reversible contraception, but neither one of those are good for cats. Right. So we are really desperate for a reversible contraceptive for cats. And the second is that pregnancy is not curative or preventative for pyometra. And you can actually develop a pyometra while you are pregnant, or while, I mean, not you or I, but I mean a cat or dog. So I just wanted to clarify that for you.
Yes,
and
no,
anybody
I guess,
else.
right, yeah, no, I guess I misspoke. I should have said it's quite curative. I find oftentimes that if you can kind of reset things, they tend to do a lot better. There are not in America, unfortunately, anti-progesterone agents available, but in Europe, for example, they really like using Alezine to treat pyometras, and I think they have really good success if they treat the pyometra and it resolves, and then they actually breed the animal again. That, again, I understand not 100%, and oftentimes once you start getting that cystic endometrial hyperplasia, you're kind of going down that pathway. But we just find our cats that have had litters. We certainly have cats that have had litters at crew as well that eventually develop pyometra, but in general, having a pregnancy or two, it seems like the reproductive tract is a lot healthier with time. So yes, thank you. I will modify that statement. I agree with you.
So why don't you hang on a minute? Yeah, I have a question for Lindsay while she's up here. You didn't mention, I don't think, unless I missed it, where you got your cats, your source?
Oh, right. So yeah, I should have included that. So for us, we do use specific pathogen-free cats. Laboratory cats, they come from Liberty, which to my knowledge is about the only game in town anymore. So they fall under the same purview of USDA in the different categories and animal dealers. So they are also inspected and have to go through the same rules and regulations. And again, you can look up their inspection records as well. I would love for us to be able to use random source cats. I think Amy did a great job explaining some of the issues with that, and for us, the biggest one is just disease management. We are set up nicely at crew. We do have a quarantine area, and even when we get cats from Liberty, we do put them through a quarantine period. There are certain diseases we're always testing for, but cats have so many diseases that can hide, that can live in the cat and that aren't going to show up during a quarantine period, that won't necessarily show up on the test depending on how we're testing. And obviously, unlike CSI would have us all believe, you can't just screen for every test, right? When you look at, so you have to pick and choose what test you're going to look for. So why I think it would be great if we could incorporate random source cats, and that would fit really well with kind of our values and what we'd like to do. It's such a risk to our cats. And it would be really devastating if we had an outbreak of herpes or another respiratory disease, because it gets really hard to keep that contained. So that's the answer.
I don't know if that's working.
Is this working? Yes. You bring up a really important point, which is when we're doing laboratory studies or these hybrid studies or even studies in pets, or out in a field condition, evaluating safety is really tricky, because as Lindsay just mentioned, some cats can potentially have disease conditions that don't manifest at the beginning. So the protocol might say we're only going to enroll healthy cats, but then it turns out that you've enrolled a healthy cat and you've treated them with your drug that you're trying to investigate the safety of, and then they get sick. And it's very hard to distinguish whether they got sick because of the treatment or whether they got sick because they were just going to get sick. So one of the tricky things in all of these types of studies is defining and following up on safety signals, because none of these can really be definitive until you get out into a wide outbred population. But maybe you want to say a little bit about how you daily observe the cats, both you and Amy, during your studies, and if you saw something odd, how you documented it.
Sure. I'll go first, since this is my life here to keep talking. Yeah, so of course it kind of depends on the study, what our symptoms are and what signs we are. For example, anything we're doing an injection, I mentioned this, we typically do daily palpation of the injection site, a visual inspection as well, but then to feel it, feeling for a mass, feeling for pain. Additionally, and especially in the beginning of any of these types of like gene delivery type studies, the veterinarian, whoever veterinarian is there that day, will be the person doing the inspection. As time goes on, as we get more comfortable, oftentimes the job of looking at the daily care falls back to the keepers. The keepers are also doing daily observations, and we actually do have a log whenever we're doing a study where they actually will record. So even if there isn't a problem, we want to have that record that every day, this was looked at, they saw nothing or this or that. And I'll give you a shout out too for helping us develop forms and keeping things on a record, as you did a good job when we started with the Gonacon study, because that was one of the first studies I started when I started Crew about coming up with a linear scale, like a numerical scale of from 1 to 4, and how to actually design a form that's informative, easy to use, but also kind of captures that information.
Exactly.
So I think that's probably.
Yeah, and we definitely followed a similar pattern. We had twice a day health checks is what we called them, and we wanted to make sure that we just put our eyes on every single cat. The cats did get closed into the building at night. They were allowed outside during the day, but we closed them in at night because of the risk of predation from owls and other birds in the area. So we wanted to make sure we had everyone locked in at night, and that allowed us to look at their eyes and just kind of make those observations. And we had a spot on the form to record that twice a day. And then we had, a medical form, adverse event form, things that we could document. If a student or volunteer was the one making the observation, they'd know to come to me, or we had a good structure set up for that. But we did twice a day observations for illness.
Yeah. And I'll just point out in the trials that are done with pet owners are instructed to record any adverse event. So it's a very odd thing, because an adverse event is defined as something unexpected that happens to that pet, whether or not you think it's related to the treatment. So even if your cat went outside and got stung on the nose by a bee, that's an adverse event, which just seems counterintuitive, because you'd think about an adverse event as being something that just might be related to whatever the drug treatment is. But the reason that the FDA and other regulatory agencies ask for this is there's really no telling what your drug might do. And so the example that I like to use is the original studies of ivermectin, where in the original study population, there was one collie that was enrolled, and that collie had a seizure during the trial, and it was recorded as an adverse event, but nobody thought it was related to the drug. But then once ivermectin, which was really HeartGuard, which was a heartworm preventative, got on the market, people started calling in that their collies were having seizures when they were treated with this drug. And then the FDA went back and looked at that original clinical trial and said, aha, there was a signal here. This was related. And it turns out that collies have a different way of processing ivermectin that does influence their brain function, and they can't take the same dose as another dog. So my point is just that sometimes, even if you don't think it's at all possibly related, why would a dog seizure from an anti-parasite drug? The records of all those studies are really critical, because in smaller populations you might not see a safety. You might not define a safety as an issue, but you might see a signal. And particularly in these early studies where you have very small numbers, the opposite tends to happen. You might see an animal suddenly develop a kidney problem, and you think, oh my God, it's from the treatment. Well, it turned out to be random. So understanding the safety signals and how you manage that in your clinical trials, and then when you get out into field trials where animals are in a real world situation, all kinds of crazy things can happen. And so trying to get your hands around that safety signal in a field situation is even more difficult.
Yeah, Dr. Pepin.
Yeah. Dave Pepin from MGH, just so I can see you, Linda. So I have a question for you. So your point about the difficulties in making a long-term contraceptive trial are well taken. So I wonder, what are your thoughts on what would be clinical endpoints that would be acceptable by the FDA to show efficacy? And can we rely on surrogate markers other than live birth?
It's a really, really tough question. I will tell you that 10 years ago, I argued with the FDA about using testosterone as a surrogate marker, and they really resisted it. They wanted breeding trials. And I said, well, we would agree to breeding trials if the FDA would agree to adopting all the puppies. But that program didn't go forward, so we didn't really ever get to a final negotiation. Back then, there were very few. Even the human side of FDA wasn't accepting surrogate markers. Now, well-validated surrogate markers are used routinely in human regulatory decisions. So I hope that you could argue for surrogate markers. You could look at testosterone levels in the males and estrogen levels, or vaginal smears in the females, and you wouldn't have to do breeding trials. But that would be a negotiation you'd have to have with the FDA. Good question.
Yeah, and along those lines, as you've been going through more of these points, something that keeps occurring to me is how much this adds to the cost, the final cost of developing these products. You made one cost statement during your presentation. Do you want to elaborate a little bit more on that?
Yeah, I'm asked a lot by everybody from venture capitalists to big companies how much it costs to develop a companion animal product. And I generally say the cost is between $10 to $12 million, and it takes between 5 and 7 years. And that's for a drug like a pain drug or an antibiotic or something that's, fairly common. For something like this, the cost is going to be significantly more because the trials, including the safety trials, will have to be much longer. And there'll be a component of studying the human exposure that's going to be really important. When I did a back-of-the-envelope calculation for the Michelson Present grants, I said about $20 million per species and probably about 12 to 15 years. So it's a long road. I mean, I think you might be able to be shorter for the rolling claim of 1 year and then 2 years and then 3 years. It might, you might be able to get that in a 10-year or so period. But the other thing that takes a long time and we haven't talked about is the manufacturing and defining the manufacturing. And I think that's also going to be an issue. But I don't think it's unrealistic to say the first one will be very expensive and the second one will be significantly less expensive as the FDA gets more comfortable with this kind of work.
I'm tempted to ask you to continue talking about the manufacturing aspect, but we've got some people waiting to ask questions, so let's switch over here. So, yeah, next.
My name is Sonia Richter. I'm a veterinary student at Western University in Pomona, California. My question is for Amy. I know you guys were doing behavioral observations of the cats and taking into consideration the social dynamics of free-roaming cats. I know you said that although you rotated the males and they were never in the enclosure together, do you think that things like urine marking could have had any impact on if they were going to mate with certain females or not?
Certainly. Yeah, we didn't. When we wrote our article, one of our lessons that we expanded on was, maybe next time we'd like to involve a skilled behaviorist to kind of look at those more nuanced aspects of how having that closely confined colony, because yes, it's sort of an intermediate towards a free roaming colony, but they're contained. They can't get away from each other, right? So we have these males and they're spraying, and maybe in a real situation, you'd be able to space out, and cats are so spatial and they timeshare, and they couldn't necessarily do that here. So definitely, who knows? It's a great question. I would expect that certainly the urine marking and the behavior impacted all of it, but we didn't have a specialist to look at that with us.
Sure. Okay, thank you.
Mm-hmm.
My name is Dale Anderson, and I've been attending the field science track, so please forgive me if this has been addressed or if it's off topic. I'm curious about IUD devices for dogs. Specifically, the ACC&D wrote a position paper on the product Dog Spiral, and I understand that there are safety concerns and there isn't enough verifiable data or studies to evaluate it as an option. My question is there support for further research into developing a safe, effective IUD?
IUDs weren't covered by this particular panel. I don't know if there's anyone in the audience that has that kind of experience that they could speak to that. Maybe Linda has something.
Well, we were part of the group that wrote the position paper on the dog spiral for ACC&D. As far as I know, it's not the type of approach that the Michelson Present grants would fund, so I don't know of any other particular source of funding unless perhaps the Morris Animal Foundation or some other group might be willing to fund it, but I don't think it's the type of approach that the Michelson Present grants are supporting at this point.
In the process of exploring that and writing the position paper on it, we contacted a number of theriogenologists in Europe and all over, and we couldn't get anyone to touch it in terms of doing more research. And they had some very good and valid reasons and valid concerns about the humaneness of inserting that device, which was concerning for us. And we still kind of kept pushing it to say, would it be possible to get more data on it? But that was just kind of the general sense of the experts, and we have to respect that. And I'm a little far from all of our, but I know we carefully wrote that paper, so that's what we think of that paper.
Yeah. Does anyone have anything else to add? Oh, here, over here.
Yeah, Steve Metcalf. I'm a veterinarian. In answer to your question, Pat Wright published this a number of years ago. They were putting very small amounts of silk into the uterus, and they all developed pyometron. And at that point, everyone just left it alone. Okay, so there's a real problem, and this is not the typical pyometron. It goes back to some of the work that was looked at using the trophoblastic reaction, but at that point, everyone just left it alone. We can talk about that later. I have a question to the panel, and this bugs me. We've been hearing a lot about both the gene therapies, the antibody responses, and the two questions that I really have is that at what point does your responsibility end? Okay, so you've adopted the cats out. Is there any callback mechanism? Is there anything that is in place so that if these cats start developing problems 3, 4, 5 years down the track, you know, how do you get around that? Because we don't know. We think we know, but we don't know what's going to happen.
Yeah, I would say
So you're referring to like long-term safety concerns,
Absolutely.
not just that this is not a good fit for this household.
So are we going to, are you going to get those cats back when they eventually pass away? You've got to look at their brains, their pituitaries, their hypothalamuses. Is there any mechanism for that? What happens if these cats start getting unwell? You know, is there a mechanism that's in place or required
Right.
to follow that through?
So for us, like irrespective of the particular study, whenever we adopt out cats, they generally get a record of all the history, so they're kind of aware of, you know, people really actually knowing, like, what did my cat help with? What did my cat? And so I think especially, again, we haven't actually adopted out any of these gene therapy trial cats yet. And when I spoke earlier in the email, it actually came from the FDA. The big thing was making sure there's a mechanism in place for follow-up. And so we do always ask, no matter what, that they kind of, the owners keep in touch with us. I think we'd have to come up with how we're going to do that more stringently for the cases like this, to follow them out long term. But we do always ask, please contact us, let us know if anything's going on. Or it works out really well for like the Gonacon study when you just make everyone that was a co-author on your on the study adopt the cats out. Right. It's a lot easier, and I can just call Linda up and ask her how her cat's doing. So.
Odie's lumps, she was the lumpiest of the cats, and she completely resolved. So, but it's a good question. And in terms of regulated studies, clinical studies, the FDA requires what they call pharmacovigilance. So any animal that gets treated with the drug, the sponsor has to provide a 1-800 number or some mechanism that they widely publicize so that people know that they can call the FDA and report an adverse event, and then the FDA will follow up directly with the sponsor. So there is a mechanism for collecting all those data, and they monitor the pharmacovigilance quite tightly the first year because it's the first time it's out in a big population, and then there's an annual requirement for reporting of adverse events. And owners and veterinarians will call and say, my cat developed cancer or developed kidney disease, and then it's up to the FDA and the sponsor to look at the epidemiology of those reports and decide whether or not they're actually related to the treatment. So there is a pretty robust follow-up for all these treated animals.
Thank you.
State that
It's a good question.
And just to follow up quickly with the Gonacon study, we followed much the same format that Lindsay described, with making sure we had contact information with the adopters and an assurance that they would reach out to us.
So I received a signal that we're at the end of our time, and I'm sure you all want to get to break. But before you do break away, join me in thanking our presenters today.